So let's talk about genes and proteins, but first let's go over some of the information we covered way back when at the beginning of the semester. So proteins, we've all seen this chart before. Proteins are made up of amino acids. So remember, proteins are a polypeptide. They're one of our macromolecules. Remember, we have 4 of them, so proteins are one of the 4. Proteins are polypeptides. They are made up of amino acids linked together by a peptide bond. Okay. So each amino acid, remember, has different properties. As we link these together, the amino acids are going to interact in the primary, secondary, tertiary, quaternary structure. And then our protein has this big 3-dimensional structure, and it has different, we can make proteins with different jobs, like we can make enzymes, we can make transport proteins, we can make proteins that just give something color, for example. So proteins are very important. It's the main thing that our cells create, and we do it, or our cells create these proteins by expressing DNA. So they get the instructions to make proteins by our DNA, which is, as we know, in our cell's nucleus. Okay, so proteins come from DNA. So let's go over DNA and RNA really quick. So DNA and RNA, remember, they're nucleic acids. They are also one of those macromolecules that we've talked about way back when at the beginning of class. DNA, double-stranded helix. RNA, single-stranded. It might be a helix. It might be a weird shape, like a cactus or something. Sometimes it folds on itself. DNA made up of 4 bases, C, T, A, and G. RNA made up of 4 bases, but it's C, U, A, and G. The sugar backbone is a little bit different, but they still have that repeating phosphate to sugar backbone, if I can draw, so on and so forth. So, and this would be our base out here. Base, okay. So DNA, RNA, DNA, the backbone that DNA uses is a little bit more stable than RNA's backbone, so that's why they differ. DNA stays in the nucleus and RNA can leave the nucleus, so RNA is like our little messenger. Viruses can have RNA in them. They could also have DNA in them, just kind of depends on the virus, but that was just kind of extra information. Okay, so DNA, RNA, hopefully we remember what they are, what they look like, what they're composed of, the basics. Okay. So we've also talked about this term, the central dogma. I brought it up last week. The central dogma is DNA makes RNA, and then RNA makes proteins. So it's expression of your genes. So DNA to RNA, RNA to protein. So remember, your DNA is made up of 46 chromosomes. It's the same in every cell in your body, aside from your gametes, so sperm and egg. Every other cell has the same genetic code. In each cell, genes are going to be activated, which are transcribed into RNA, and then the RNA is translated into a protein. So that protein, remember, it could be like an enzyme, it could be just like structural, it could be functional if it's like a cell membrane transporter or something. Remember, we have a lot of proteins in our like mitochondria. We have a lot of proteins, well, we don't in chloroplasts, but proteins, big part of your body. So your cell makes them all the time, and that's the result of your genes being expressed. So I like this meme down here. It says, "Crush, I don't like you, you're ugly. My gene, no." So central dogma, make sure you commit this to memory, DNA to RNA, RNA to protein. Okay, so the first step in the central dogma is transcription, and then the second step is translation. We're going to come back to those in a little bit, but just keep those kind of in your mind. Questions so far about proteins, questions about nucleic acids, central dogma, what a crush is? No? Okay, cool. All right, so let's look at the central dogma a little bit more. You have 46 of those. Let's pretend, okay, so let's look at flowers, I guess. Flowers have this particular pea plant, has 7 chromosomes, oh, oops, 7 chromosomal pairs, okay. So on one of its chromosomes, it has a gene for flower color. So remember, last week we learned that each chromosome carries more than one gene. That was one of the experiments that we talked about, that each chromosome carries one gene. So that was visualized by like crossing over, how different variations can come about, so on and so forth. So one of the genes might be like this. That representation here is highly unlikely. Genes are not usually this short. Usually genes contain like a few thousand to a few million base pairs. So this one contains what, 3, 6, 7, 8. This one has 8 base pairs, highly unlikely. So a few thousand to a few million base pairs is normal. A few thousand to a few million base pairs. So that sequence will be transcribed into what's not shown here, into RNA. And that RNA makes a protein. And that protein might make pigments, in this case, might make pigments that are purple in color. Let's see, so genes are, again, a few thousand to a few million base pairs long. One chromosome, if we're talking about humans, so let me do this. So humans, remember we have 46 chromosomes. For example, chromosome 1 has about 2,000 to 2,100 genes. So somebody do the math on that. If each gene contains a few thousand to a few million base pairs, let's see, 2 million. If chromosome 1 has 2,000 to 2,100 genes, and each gene has that many base pairs, that's a lot of base pairs on one chromosome. So your DNA is going to be copied into RNA at many points along one chromosome at any one time. So, and just that for, I guess for simplicity, we're just going to look at like one gene at one time, and we'll look very closely at the sequence, and we'll kind of leave out the extra stuff on the ends. But just so you know that the central dogma is happening all over one chromosome at any one point, and it could be happening over and over and over again. So one segment of RNA might be copied a few thousand times before DNA is closed back up. Okay. So let's look at the flow of genetic information here. So our DNA, the first step is what? It's transcription. The next step is processing, and then the final step is translation. So during transcription, again, RNA is made from DNA. We're making an exact copy? No. We're making a complementary copy, but we're making it into RNA, right? So if we're looking at our DNA here, it's not an exact copy, right? It's a complementary copy. So that RNA will be processed before it leaves the nucleus, and then it will be translated into a protein, or the beginnings of a protein at least. So this is just the basic flow of information, or AKA gene expression, or AKA the central dogma. So that's correct. C is the right answer. Transcription, processing, and then translation. All right, great job. Commit those to memory. All right, so I like to think of the central dogma like a recipe, and you can't rip the recipe out of a book, so you have to copy it down onto a piece of paper. So you can think of DNA as kind of the book, the recipe book. So, or maybe it's like a reference book that you can't check out of a library. But you can make like a photocopy of it, or you can handwrite notes out of said book. So this would be like your notes. And are you going to take notes on every part of that book? Probably not. You're probably only going to take notes on the section that you need to take notes on, so a small segment. Those notes you can take with you, and you can do whatever you want with them. In this case, you are making a recipe, or you're making food, which could be like the protein from the notes. So let's double back. If DNA is like a recipe book, you can copy down a recipe, one recipe out of a book of maybe 1,000 recipes, so one small recipe, and then you can take that recipe with you to make food, or in this case, protein. And then once you're through with the recipe, you can make the food again with it, or you can throw it in the trash. Okay, so what the RNA here can do, what I'm trying to show you, is that it starts in the nucleus, close to the DNA, but it can leave the nucleus. The DNA cannot leave the nucleus. It has to stay. So that's why we need RNA in this case. We always need RNA because DNA cannot leave the nucleus. So RNA leaves the nucleus to go make the protein out in the cytosol of the cell, or where it can access a ribosome. So it might be in the cytosol, it might be on the rough ER, right? Those are two places we can find ribosomes. So DNA can't do that, so that's what we need RNA for. Okay, look at this image here and try to fill in these letters with words in the top right corner. So let's move on then. Okay, so these are the three types of RNA. So there's rRNA, mRNA, tRNA. So mRNA, there's supposed to be little GIFs playing here. But we already labeled, oops, wrong one. rRNA, let me get my pen back, there we go. So rRNA is a ribosome. It's called ribosomal RNA. And ribosomal RNA has two units. There's a small and a large subunit that make up a ribosome, and each of them like clamp onto the messenger RNA. So the rRNA is what is going to catalyze the protein creation, or it's going to catalyze the reaction between amino acids to make that protein. So that's one type of RNA. Another type of RNA is messenger RNA, which we're going to practice creating in quotes today. Messenger RNA is the transcript that ribosomes use to create that protein. So that's copied from DNA, right? And then transfer RNA are those weird shaped ones. So they look like this. Oh, that's a terrible picture. But anyway, they look like that. They have those three codons attached to them, right? And those anticodons will match to codons on the mRNA. So you have 64 types of tRNA in your cells, and each of those 64 types has an amino acid on it, just one amino acid, and so that's what brings the amino acids to the ribosome, which helps create our protein. So let's see, all three of these RNAs are in translation. They take part in translation. The only RNA that takes part in transcription is mRNA. So ribosomal RNA and tRNA are only going to be found out in the cytosol of the cell. You may be able to find mRNA in the nucleus as well as the cytosol. Okay, so let's talk a little bit more about amino acids and codons. So this is a codon chart. You've probably seen one before. You may have seen one that's a circle. Either of the two work, but we're going to use the square one for right now. So this codon chart shows you all the possibilities of codons. So I already told you we have 64 possible codons. And those codons code for amino acids, which are abbreviated by 3 letters. So remember, a codon is made up of codon equals 3 mRNA bases. Okay, so 3 mRNA bases is a codon. So like GUU is a codon. And that codon will code for a tRNA anticodon, which brings over this amino acid, which is valine. So if I gave you, let's see, Let me give an example here. Let's do like AAU. So if we have a codon AAU on our messenger RNA, our ribosome is going to read it starting with the first letter, which is A, which would be somewhere on this row. So first letter A, somewhere on this row. Second letter's A, so somewhere on this column. So it would be at the intersection here. And AAU, the third letter is U, so we can find it easily right there. ASN, which I don't ASN. I have to think about which amino acid that codes for. Let's look. ASN is, I think it's asparagine. Yeah, that's ASN. Okay, so that codon, AAU, codes for amino acid asparagine, ASN. Oh, no, maybe it's not asparagine, because that one's ASP. I'm not sure what ASN is. Anyway, it codes for this one right here. So each of those codons, again, they code for an amino acid. What you might see are some special amino acids, like stop and start, or MET in this case. So those special codons, the start codon is AUG. Codes for methionine, the amino acid methionine. So you might have a sequence like this. The ribosome is going to look at this sequence on mRNA, and it'll read it, but it will not start creating a protein until it gets to that codon right there that codes for methionine. So everything that comes before the start codon is just kind of nonsense. So you have to have a start codon in order to start creating a protein. Methionine is the only start codon, AUG. Stop codons, you don't necessarily need a stop codon, but it's nice to have one. There are 3 different types of stop codons: UAA, UAG, and UGA. They don't actually code for an amino acid. So they just tell the ribosome to detach from mRNA, and that makes the protein complete. So if we had a sequence, let's see, let's put U, A, G. Oh, I can't fit G. Well, let's see. We'll do AUG, so that's our start. And we'll do like A, C, G, A. And then we'll do U, A. And then we'll do like some random stuff. G. Okay, cool. So we'll start at AUG. We'll make methionine, and then we'll make another amino acid and another amino acid. And then we'll get to UAA. So that's our stop. So our ribosome will detach from mRNA, and everything after stop will not be coded for. So that's a problem if there was like a mutation that created a stop codon instead of an amino acid codon. So that would be a big problem. It would make our protein really short and probably not functional. That would be an issue with the stop codon, but a good thing is that if extra stuff got copied, it would not really matter, right? If we had a stop codon. Okay, so let's practice translation here. And then that's where we'll stop today. Okay, so if we have sequence AUG, we already said that would be methionine. And then we had UAA, CGU, AA, GC. So UAA. U, first letter U, second letter's A, UAA is stop. And so since we reached a stop codon in this sequence, we wouldn't code the rest of the sequence. It just wouldn't happen. A stop codon like UAA, UAG, or UGA causes the ribosome to fall off. Now, let's say we didn't have a stop codon, we would keep coding. Notice we have a fragment of a codon down here. Fragments of a codon do not code for amino acids either. So if we just have one letter, we wouldn't code for anything. We have to have 3. Okay. And then I think I accidentally erased it. Yep, I wish it didn't erase. And I switched slides. But anyway, are there any questions about the codon chart? No? Okay. One last thing I want to tell you before you go is that you probably noticed, without me saying, that there are codons that code for the same thing. So let's look at serine. So serine here, 4 codons here code for serine, and then there's 2 down here that also code for serine. So out of the 64 possible codons we could make, there's 6 that code for the same amino acid. So you might be asking, like, why do we have 64 codons and only 20 amino acids? 20. So since we have 20 amino acids, 64 codons, mutations that occur in DNA or mistakes that occur in RNA can still code for the same amino acid. So they won't be so big of a change, right? So if we had a sequence, like an RNA sequence that was like UC, U, A, and something happened and now it's UCC, A. These 2 codons in the front are still going to code for serine. So the sequence may have changed, but the amino acids that we code for haven't changed. So no big deal that there was a mutation. So most of the time when there's a mutation, like a point mutation like this, not a big deal. So this is called degeneracy. So if we have degeneracy or we have changes in our sequence, it's not a huge deal. Also, the genetic code is universal. So every organism that's living on Earth has the same codons and it has the same amino acids. So UUU codes for PHE or phenylalanine. So phenylalanine, UUU, if we put this codon in a bacteria, it'll code for that amino acid. If we put this codon in a horse, it'll code for that amino acid. If we put this codon in a fish, it'll code for that amino acid. So we can put like a gene for insulin in a bacteria, a human gene for insulin in a bacteria, and that bacteria is still going to create insulin because it uses the same code. Okay. All right. So I don't want to go over, so we're going to end it here on
all right, so RNA is made from our DNA template. So DNA splits open, we make RNA, and that's translated into a protein. So let's look at this transcription process first. So remember, the Central Dogma has 3 steps. We have transcription, processing, and then translation. So we're going to talk about each of these steps in more detail today. So let's start with transcription. There are 3 steps of transcription. There are also 3 steps of translation, and they are both named the same, so that kind of helps to remember. The first step is initiation. So remember, transcription, we try to remember that transcription is the one that comes first because it is TRANSC. TRANSL for translation, C comes before L, so transcription is first. That's how we remember it. So transcription starts with initiation. So if we're looking at, we're going to use prokaryotic DNA here for our example. So on prokaryotes, their genes are surrounded by -10 and -35 regions. There's one that's called the Tata box, which you might be familiar with, and that is just a promoter. So that tells RNA polymerase that, hey, this is where the gene starts. Okay, so at the Tata box, TATAAT. And then there's also another region further ahead of that, so -35 ahead of that, and that's TTGACG. That's not easy to remember. So Tata box comes before the gene begins, and so that's part of the promoter. These numbers, -10, -35, is how many nucleotide bases it is from the beginning of the gene. So this T starts -10 away from the gene, or 10 nucleotide bases away from the gene. And then this T starts 35 nucleotide bases away from the gene. Oh, I don't think you can see me pointing. But anyway, so that T on the Tata box starts 10 nucleotides upstream from the beginning of the gene, or the beginning of our transcription site. And then this T on the other promoter region is 35 nucleotides ahead of the transcription site, or ahead of the gene. So you can see RNA polymerase, this tan thing, is going to attach to that promoter region, which kind of situates it correctly for the gene to begin, or for the gene to be in the right spot on RNA polymerase. So that's why the promoter is there. 1, it's an attachment site for RNA polymerase, and 2, it tells it where the gene begins. So that's initiation. So the second and third step, elongation and termination. Elongation, simply just, transcribing mRNA. That's all it's doing. So RNA polymerase is going through, reading the DNA template and making the mRNA template. And then termination will happen once there is a termination zone. So just like a promoter region, there will be like some region downstream that would be a termination zone. And once it reaches that termination zone, RNA polymerase cuts off mRNA from it, and then RNA polymerase detaches from DNA. Okay, so initiation, elongation, termination for transcription. Let's watch a video that goes over what I just said. Let's see, I think I have to share my screen.
Proteins are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. Motor region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other.
Okay, so do we understand it a little bit more? Hopefully that visual helped. But you could see that the RNA polymerase enzyme, right, it's a protein, it's an enzyme, is going to split DNA apart and just build that template, or build that mRNA off of the template. So they did mention those promoter and terminator regions, so don't forget about those. Promoter is upstream from the gene. Terminator is downstream from the gene. That's the terminology that they like to use. So once that terminator region, of course, is reached, then transcription stops. So transcription, remember, is the first step in our central dogma. Transcription's main job is to make that mRNA. So once we make mRNA, we can take that to make the protein, but something has to happen before we can use it to make the protein. I do want to remind you that prokaryotic and eukaryotic transcription, translation is a little bit different. In bacteria, transcription and translation happen like instantaneously from each other. So once that mRNA starts to be transcribed from DNA, like immediately a ribosome attaches to mRNA and starts translating it into a protein. So mRNA is still being built here, but ribosome is already making a protein out of it. In us, in eukaryotes, it takes a little bit longer for transcription translation to occur because we have a nucleus, and mRNA is really the only other thing other than DNA allowed in the nucleus. So we also have some like extra things in our DNA, like extra base sequences, and we have to eliminate those from mRNA before we can make the protein. So that's what our processing step is for. So remember, we have 3 steps: transcription, processing, and translation. Let's talk about this processing step here. You can notice already that this mRNA that we've made from DNA in transcription looks a little different. We've added some things on the end and eliminated some things in the middle. So mRNA processing, wait, let me go back, does occur in the nucleus. So it has to happen before mRNA can leave the nucleus and enter the cytosol of the cell. Okay, so processing and transcription occur inside the nucleus. Okay, so mRNA processing, let's see, I'm going to go to this one. What does it do? So first, our mRNA will look like this up here. So our primary RNA transcript. So this is our mRNA. So you can notice it has some different sections on it, exons, introns, and so forth. So what do those things mean? An exon is the good part of the gene. So that's the segment that is actually going to code for amino acids. Okay, so the exon is the part that we want to keep. Now we want to get rid of introns. So those are just like nonsense between genes, or it could be that introns code for something, but we don't need them in this protein, so we're going to get rid of them. Okay, so introns we keep, exons we keep. I like to think of it, exons exit the nucleus with RNA, right? So introns, we get rid of them. Okay, so exons kept, introns are deleted. Some other things that we add is a 5' cap. A 5' cap is a guanine cap, methylated guanine cap, and I know that probably doesn't mean much to you, but this cap is needed on our mRNA for recognition by our ribosome. So our ribosome isn't going to recognize our primary mRNA transcript up here because it doesn't have that cap. So if we put a cap on it, our ribosome is going to recognize it as mRNA. We also add a poly-A tail, so it sounds just like it is. It's just a bunch of A bases added to our mRNA, and that just helps prevent breakdown or like stress or degradation to our mRNA template. So just extra protection. So the 5' cap does add some protection, but its main job is to be recognized by our ribosome or our RNA. Okay, So processing, let's see. Yeah, so processing, we get rid of our introns, we keep our exons. It's called intron splicing. And then we also add our 5' cap and our poly A 3' tail. So 5' is the end of our RNA, and it's the front end of our RNA. 3' is the end of our RNA. Okay, so let's watch our video again. I paused it at, I think, the right spot. Let's see. All right.
The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns. In order for the messenger RNA to be used in translation, the non-coding introns need to be removed, and modifications such as a 5' cap and a 3' poly A tail are added. This process is called intron splicing and is performed by a complex made up of proteins and RNA called a spliceosome. This complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can leave the nucleus through a nuclear pore and enter the cytoplasm to begin translation.
Okay, so let's do, I have 2 poll questions here. Let's see, so which pre-mRNA processing step is important for initiating translation? So the correct answer is D, the 7 cap, the 7-methylguanosine cap, or the 5' cap. So in order to initiate translation, we need that cap. So remember, translation happens on the ribosome. For the ribosome to recognize our mRNA, it needs the cap on the end of it, or on the front end of it at least. All right, let's see. Let's do this other poll question. This one needs a little bit of math. A, B, C, and D. All right. So it says, what is the predicted size of our mature mRNA after we do some splicing? So remember which ones we keep and which ones we get rid of. And then we're going to need to add those up and see what our answer is. BP is just referring to base pairs. So like the first segment here is referring, this exon is 100 base pairs long. So that's all the BP stands for. So we're seeing how many base pairs long our mature mRNA is. We've got a few answers in. There's 3 more people, 2 more people. Looks like most of you got the right answer so far. I'm going to go ahead and show. So most of you said B, 295 base pairs. That is right. So our exons, remember, are what's going to exit the nucleus to the ribosome. So if we add them up, 100 plus 75 plus 120 would be 295 base pairs long. So good job if you got that right. You'll see a question like that on your quiz, I believe, for this week. All right. Okay, so let's get on to translation. So we've talked about transcription, which is the first step of the central dogma. We've talked about RNA processing. So let's talk about translation, so the final step, the flesh and blood step, right? It's literally making flesh and blood. So translation, we also call it protein synthesis, since we are making a protein. And what is actually happening is we're decoding our mRNA sequence and translating it into a protein. And this occurs on a ribosome. So remember, ribosomes, they are a form of RNA. We call them rRNA. And they are made up of two parts. And those two parts are called subunits. Let's see if I have a picture of one. Yes, okay. So subunit 1 is very small, or subunit A, or the small subunit, whichever you want to call it. And then there's the large subunit, or subunit B. So the small subunit is going to attach to mRNA. So that's actually what's going to recognize that 5' cap on our mRNA sequence that we just put on it. So once the small subunit recognizes that 5' cap, it attaches, and then the large subunit will then attach to that conjunction there. So remember, our sequence doesn't begin, or it doesn't begin coding until we reach AUG. So remember, AUG is our start codon, codes for only one amino acid, and that's our initiation step right here. Okay, so mRNA attaches, it recognizes that 5' cap, the large subunit attaches, and then we start where it is, where it says AUG. So if there's anything before AUG, it does not get coded. We need that start codon first. Okay, so our next step, and I don't know why it says 1 again, but number 2 is elongation. So remember, I told you initiation, elongation, termination are the 3 steps for transcription and translation, so that's easy to remember. So translation, elongation, we're just elongating that protein. So as new codons are recognized on our mRNA, tRNA will come in with the associated amino acid, and that amino acid will attach to the previous amino acid, making our polypeptide. So our polypeptide is our protein made up of amino acids. Remember, amino acids are not just the R group. Let me see. Here we go. So this is an amino acid. Let me see if I can circle it with my mouse. So that's a whole amino acid. The part that we typically refer to is just that right there. So there is another portion of an amino acid, but it's the same no matter what amino acid we talk about. So if this is like methionine, this would be the only part that changes. Or if this is like phenylalanine, this would be the only part that changes. The rest of it stays the same. It's kind of like our base pairs in DNA. So we only refer to the base pairs, but we still know that DNA bases have a sugar and a phosphate component attached to them. We just don't talk about them because it's the same for everyone. So the R group is the only thing that's changing on these amino acids. The way that we link amino acids together is by a peptide bond. So let's say that this is our methionine here, and then this is our next base, or like our next codon that codes for this amino acid. We're going to link them together by the peptide bond. And that peptide bond is catalyzed by the ribosome. Okay, let's go back to the slide. So that peptide bond here, let's see if I can circle it or point it or something. Right there. That's our peptide bond that links our amino acids together. So you've probably noticed that there's some weird letters on the ribosome, E, P, and A. The important part of the ribosome, or the important slot of the ribosome, is the P group, or the P slot. That's where the hard work is happening. The E and the A are just kind of like exit and entrance points. So the P is the slot where all the magic is happening. So as, I guess if we go in order, this should be a piece, or this should be the A slot up here. So this would be A, and then it moves over to the P slot. Another one comes into the A slot. This one moves over to the E slot. This one moves over to A, or to P from A, so on and so forth. Okay, so our last step of translation is when we reach the stop codon, so that's termination. Our stop codon, remember there's 3 of them. There's like UAG, UAA, and UGA for our stop codon. And once we reach the stop codon, the ribosome detaches, the protein is made, and our mRNA can be either recycled or eaten up by like a lysosome in the cytoplasm. So when we reach the termination step, our protein is complete. It's hopefully properly folded in like the 3D structure that we typically associate with proteins. I like to think of the tRNAs that we're looking at here, so with the UAC and the amino acid attached to it, as just bringing amino acids to this large overbearing polypeptide chain. Okay, so remember, tRNAs are definitely an important part to translation. They bring over amino acids, and there are 64 different tRNAs in your cell. And there's 64 because we have 64 codons. So then there's 64 anticodons that we have to match with our tRNA. All right, so let's take a look at this video to summarize what we just went over. Let's see. I think I need to find it first.
The job of this mRNA is to carry the gene's message from the DNA out of the nucleus to a ribosome for production of the particular protein that this gene codes for. There can be several million ribosomes in a typical eukaryotic cell. These complex catalytic machines use the mRNA copy of the genetic information to assemble amino acid building blocks into the 3-dimensional proteins that are essential for life. RNA copy of the genetic information.
Okay, I wanted to stop the video right here because it might not be apparent what you're looking at. This long strand, you probably could have guessed that's our mRNA. This in the middle is our ribosome. And you can see there's like slightly different colored blues. So the small subunit is this light blue on the bottom. The large subunit is this darker bluish purple on the top. And then coming out of the top of the large subunit is our protein that's being made. And each of these things that are floating around, let's see if we can get a better view of them. Maybe not, but each of the things that are floating around are tRNA, and they have a little red dot on them, which would be the amino acid that's building our protein. So once those tRNAs come in, they're going to show you what actually goes on. Let's hit play again.
Use the mRNA copy of the genetic information to assemble amino acid building blocks into the three-dimensional proteins that are essential for life. Let's see how it works. The ribosome is composed of one large and one small subunit that assemble around the messenger RNA, which then passes through the ribosome like a computer tape. The amino acid building blocks, that's the small glowing red molecules, are carried into the ribosome attached to specific transfer RNAs. That's the larger green molecules, also referred to as tRNA. The small subunit of the ribosome positions the mRNA so that it can be read in groups of 3 letters known as a codon. Each codon on the mRNA matches a corresponding anticodon on the base of a transfer RNA molecule. The larger subunit of the ribosome removes each amino acid and joins it onto the growing protein chain. As the mRNA is ratcheted through the ribosome, the mRNA sequence is translated into an amino acid sequence. There are 3 locations inside the ribosome, designated the A site, the P site, and the E site. The addition of each amino acid is a 3-step cycle. First, the tRNA enters the ribosome at the A site and is tested for a codon-anticodon match with the mRNA. Next, provided there is a correct match, the tRNA is shifted to the P site, and the amino acid it carries is added to the end of the amino acid chain. The mRNA is also ratcheted on 3 nucleotides or 1 codon. Thirdly, the spent tRNA is moved to the E site and then ejected from the ribosome to be recycled. As the protein synthesis proceeds, the finished chain emerges from the ribosome. It folds up into a precise shape determined by the exact order of amino acids. Thus, the Central Dogma explains how the 4-letter DNA code is, quite literally, turned into flesh and blood.
Okay, so hopefully that helped sum up what we just went over. Let's see. We're going to put the steps in order here in just a second. The steps of translation, at least. All right. So last thing that we're going to look at today, we're just going to put these steps in order. You'll see a question like this on your quiz, so that's why we're going to go over this a little bit more in depth. So J through N are the steps, and no, they're not in the correct order right now. We do need to put them in the right order. Okay, so do we agree?